Complement system. This is a session that is going to focus on one very important pathway that the complement system has adopted, and that is the alternate pathway. And also, the regulation of alternate pathway will be looked at in brief. Now, the kind of regulation that is present for alternate pathway, one finds a similar kind of regulation, or basically the same kind of regulation is applicable both to classical complement pathway as well as to the MBL pathway per se. So let us look at the learning outcomes of this session. The alternate complement pathway is antibody independent pathway, unlike the classical pathway, which is highly dependent on antibody antigen complex. Although it is not always necessary that the classical complement pathway needs an antibody to bind. What has been observed is that the initiator molecule of the classical complement pathway, which is the C1 complex, can recognize more than 100 different pathogen associated molecular patterns. But it does very effectively bind to the FC region of the antibodies that are bound to an antigen. Now, the alternate complement pathway also is an important component of the innate immune system, with the C3 component in the serum initiating the cascade to assemble finally the membrane attack complex. So whether it is the classical complement pathway or the MBL pathway, or it is the alternate pathway, it will ultimately lead to forming the membrane attack complex, which is responsible for the lysis of the cell. The C3, after cleaving to C3b, is able to recognize, so the C3b is able to recognize and bind to molecular patterns on the pathogens, thus forming opsonins. So when it binds to different pathogens on the surface or to different cells on the surface, it can recruit neutrophils, or it can call in macrophages to phagocytose the cell that it has bound to. The alternate pathway is permanently active. Now, this is interesting. Unlike classical complement pathway, which was active only when it is binding to an antigen antibody complex, in this case, the alternate pathway is permanently active at very low levels. Now, the question arises why? It's because C3b is able to recognize the molecular patterns on various different antigens. So, therefore, in a way, the alternate pathway is responsible for immunosurveillance, that is, they are on the lookout for a foreign particulate matter, and that is by the C3b being able to bind to different molecular pathogen associated molecular patterns. Now, in physiological conditions, the alternate pathway is considered to be a dominant pathway. So C3 is generally converted to C3H2O. So basically C3 is going to bind to a nucleophile, and generally the nucleophile is H2O. Now, how is this formed? So what happens is there is an internal thioester bond that is present in the C3 that is hydrolyzed by water that is present in the the plasma. So you can very well see that it is a spontaneous hydrolysis that is happening, but this hydrolysis is slow. Now, these are therefore considered to be C3b like, or they seem to have C3b like properties. But on the hint side, if you were to think, then this is not a very strong initiator of alternate pathway. If you have C3b, C3b is more strong and an initiator of alternate pathway. So, but what is very interesting to note is that in physiological conditions, when we are saying that the alternate pathway is dominant and that it is permanently active, okay, this is what we mean, that the C3 is going to always get spontaneously converted to this form. So let us understand how and when you have basically C3 component, okay. If acted on by C3 convertase, C3 convertase is going to remove the C3A. So this is the C3A that is going to be removed, so cleaved off, and what is going to remain is C3B. So you can see how the conformation of C3b is very different from C3. So this domain, which was earlier present over here, is now present here. So you can see how the conformation of the C3b changes, and you also would notice therefore that the C3b is able to bind to the membrane or the surface of certain cell. Now, when it is spontaneous, the C3 is getting converted into the C3H2O form. But here what you observe is that the C3A is not cleaved off, okay? And it remains attached, but there is a conformational change that has definitely happened with the binding of the H2O molecule. So here again what you can see is that the C3H2O has a very similar conformation as the C3b has, and therefore it is suggestive of the fact that it has C3b like properties. Okay. Now C3b itself can definitely bind to pathogen associated molecular patterns, and therefore C3b is considered to be a molecule that is carrying out immunosurveillance because it is able to identify a lot of pathogen associated molecular patterns, and not just identify but actually bind to it. So another another concept that is being debated on and that is being studied is that does the C3b also get converted to the C3H2O form itself? The question arises, or the question arises because, like for example, when the C3 convertase forms C3, converts C3 into C3b per se. And if you remember in the classical complement pathway, where you have the C4b2A acting as a C3 convertase, one C3 convertase is able to form about 200 or greater number of C3bs. So, what has been speculated is that many of the C3bs that are formed during activation of a classical complement pathway, not all of them would be probably going and binding to the surface of particulate matters or the surface of foreign particulates. So then what is happening to those C3bs? Well, one of the understanding, or one of the speculation, is that C3b also probably binds to a nucleophile like H2O and forms or goes into the C3b H2O form. Now, this spontaneous conversion of C3 into the C3H2O is also called as a tick over theory. Well, one reason why for calling it tick over is, like, for example, from this position it has suddenly gone to this position, and so that conformational change is what is called as a tick over theory. Now, this C3H2O, after the tick over, okay, the conformational change has happened in such a way that this part of the C3H2O is now open to bind to a certain molecule. So here is something that can come and bind. So of course, in the alternate pathway, what has been observed is that this region which is now exposed can be bound to by factor B. And in fact, the factor B, when it binds to C3H2O, it kind of stabilizes the C3H2O further. And so this C3H2O factor B complex per se together is acted on by the next component of alternate pathway, which is factor D. Now, factor D is a serine protease, and factor D can selectively cleave at the factor B region to form what is called as factor BB. So, BB is formed and the BA is released. Okay. So, now what you have formed is a C3BB, and this C3BB is also catalytically active, and in fact, it is a C3 convertase. So therefore, from C3H2O on binding to factor B, that factor B getting converted to BB, you get a form of C3, C3BB, which is now catalytically active, and this also is a serine protease. So effectively, C3 convertase is going to act on all the native C3 that is present in serum and convert it to C3A, which is an anaphylatoxin. So it will move away and in fact help as a mediator in inflammation reactions, and of course C3b is going to be formed. Now, the more the C3b is formed, the more it will bind to factor B, the more it will form C3BB. And so it is now involved in something that is called as an amplification loop. So as you have more and more C3BB formed, you will have more and more C3B formed. So there is an amplification, many-fold amplification of formation of C3B. Is it good? So the point that is to be noted over here is that this C3b is able to bind to the molecular patterns that are present on pathogens, and why therefore should it not be good. But what has been observed is, okay, before we go into that, please do understand that therefore factor D becomes one of the first components to actually lead to amplification of C3b. So it is first component of the amplification loop itself. Now, let us just look at factor D. Factor D itself is present as a zymogen. So we all know that in complement pathways, most of the enzymes are in the proenzyme form. So they're in the inactive form, and then they are cleaved to give rise to a small component and the large component. And this large component, the conformation of which is such that it becomes proteolytically active. So the same has been observed with factor D. So in case of factor D, when you have this is the factor D, so that is cleaved off. So the pro pep part is cleaved off, and what results is an active factor D, and this active factor D, as we can see already, is functioning as a serine protease. So question that comes up is here is what converts the factor D from its zymogen form to an activated form? So the answer to that is that there are many other serine proteases, and especially in this case, it is MASP3 and MASP1. So serine protease 3, serine protease 1, and this is basically mannose associated lectin binding serine protease 3, and same goes with this. So you can see how you have already enzymes. So these would be under regulation of something, a further trigger or a further signal. So therefore, the MSP3 and the MASP1 are controlling the regulation of factor D. So it is able to activate zymogen into an activated form, and we already can see that the activated factor D is able to amplify the C3b production. So if this is not active, you will not have enough of C3b formed at all. So therefore you can see how this, if controlled, you can control the amount of C3b that is formed. And interestingly, factor D is synthesized by adipocytes. Now when you look at the structure of factor B per se, you can see how you have different domains present, okay? And of the domain, if this is the part that is removed, so this is the BA that is removed. Now, the moment this domain is removed, you can see how the catalytic triad region is swirling, or is moving at an angle of at about 68 degrees to form this conformation. So in this form, the factor BB acts as a serine protease, and therefore you can see how the conformation change is very important to ensure that the factor B, only when it is factor BB, then it can act as a enzyme. If you still have this component bound, then it is in a form that it cannot act as a enzyme. So you can see that this therefore is the proenzyme form, and when it binds to C3b and gets converted to BB, then it becomes the catalytic triad. So the C3PB leads to, as mentioned, C3b amplification, because this is C3 convertase. So C3b amplification leads to formation of C3b, and that C3b can definitely go and bind to pathogen associated molecular patterns on the pathogens. They can bind to our normal cells, which is the host cell. So there are many surface molecules that are present on the host cells that can be recognized by C3b. It can also, I'll just come to that. It can also bind to apoptotic cells. So you can have on the apoptotic cells what is called as uh, um, death associated molecular patterns, or damage associated molecular patterns, and therefore you can have C3b go and bind to the apoptotic cells as well. Now, interestingly, what question that comes up is that if C3b binds to a pathogen, well and good, because it has recognized a foreign particulate matter. If it recognizes an apoptotic cell, again good, because it could initiate a process of eliminating apoptotic cells, like there is a process of eliminating the pathogens. But then, why would one want the host cells to be eliminated? So the C3b on binding the host cells can trigger a pathway that can also start lysing the host cells. Now that is going to be detrimental. So therefore, there is a set of complement regulators that can bind to the C3b on the host cell and prevent the C3b from becoming active. So the C3b is basically inactivated. Okay, now therefore we come back to another question. If ultimately you need a set of regulators to stop the C3b on the host cells to become active, to become inactive, why then should you have a system where the C3b is binding to the host cells? So the understanding is that the surfaces of most of the pathogens or apoptotic cells or the host cells, the sugar constituents are more or less the same. Okay, so the binding of the C3b, if you cannot have very specific molecules. If it is so specific in binding only to the PMPs on the pathogens, then the PMPs have to be very distinctively different from sugar molecules that are present on the host cells. Okay, so you would find that there is a possibility of having the same kind of sugar structures on the host cells also. So C3b definitely goes and binds to the host cells. So, in order that the C3b is able to recognize a range of molecular patterns on the pathogens, one has to kind of compromise with the fact that then the C3b can also go and bind to the host cell. Okay, so therefore, the need of the hour would be to somehow ensure that all the C3b that is bound to the host cells do not call in for lysis of the host cells, and that is by inactivating the C3b present on the host cell. And so you have a set of complement regulators present. Now, those that bind to the apoptotic cells, okay, so the C3b along with the initiator molecule of the classical complement pathway, which is a C1 complex, and the MBL pathway, they can together clear away these apoptotic cells. Now, coming to the feedback loop to just understand how the feedback loop is maintained, okay, what is very clear is that C3b on binding to B is acted on by factor D. Factor D will form BA and BB, and now the C3BB is a C3 convertase. But the C3 convertase itself is stabilized to keep maintaining the amplification loop, and that stabilization is carried out by another protein in the serum, which is called as the properdin. So the properdin is actually binding to the C3BB complex to form C3BBP. And this C3BBP is still a C3 convertase, but the efficacy of this is greater than this because now this structure has been stabilized. So therefore, the rate at which this C3 convertase is going to convert C3 into C3A and C3B is going to be much more than just the C3BB complex. So binding of the properdin is actually a positive regulator of the alternate pathway because it is ensuring that you are having more of C3b present. That same C3b can again go and bind to factor B, initiate the process of formation of again a stable C3BB properdin complex, and that is going to result into forming more C3b. So in all likelihood, slowly and gradually, as you have more C3b being formed, you will have more and more and more amount of C3b being formed. So therefore, that is a feedback loop that is very important. Now, also what has been observed is that there are various platforms that the alternate pathway has for its activation. So say for example, you have an activated neutrophil and this neutrophil has been activated say by, for example, an anaphylatoxin like C5a. So in response to the C5a, okay, what can happen is that several molecules of properdin are released by these activated neutrophils, and we all know that properdin is something that is going to stabilize the C3BB complex. So you can see how you have a C3BB complex, C3BBP complex formed on the surface of the neutrophil. So you can see this is a platform on which you can have the activation of the alternate pathway. Similarly, you can also have the properdin enabling the C3BB complex to bind to the platelets. One can also observe that on endothelial cells, you may have several adhesion molecules that in turn can bind to C3b, and then that C3b can be bound to by the C3bb. So you see, wherever you have the C3bb bound, now those cells are considered to be activated, and so they are called as activated neutrophil, activated platelet, activated endothelial cells. But the point that is important is that the C3b has been allowed to bind to its surface and form a active C3 convertase complex. So repeating, more the C3bb, more you have the C3b. And proper DIN is secreted by macrophages, neutrophils, T lymphocytes, mast cells, etc., thereby promoting the alternate pathway. That is important. So this is again, by the look of it, it is a positive regulator. Now, therefore, let us understand that when you have a C3b feedback or amplification loop, there is also a breakdown loop that is needed to balance it out. You cannot always keep having a high amount of C3b. So at a certain point, it is necessary to ensure that the C3b levels come back to normal. Okay, and so there is a C3b breakdown loop as well. So in let us just go through the feedback amplification loop once. As you can see over here, C3 can spontaneously get hydrolyzed to C3b. This C3b can bind to by factor B with to form C3bb complex, which can be acted on by factor D. Factor D cleaves the factor B in such a way that the BA is released and what remains is the BB. And this C3bb is what is the C3 convertase. So it is going to further act on C3 and form more of C3b. So you see how you have the amplification loop formed. Now, suppose the C3b, instead of binding to the factor B, binds to factor H. So there can be a competition of binding of C3b to factor B or to factor H, and that is important in order to maintain a balance. So therefore, the C3bb can be bound to by factor H. When the C3bb is bound to factor H, it can be acted on by factor I. Factor I is an inhibitor. It would cleave the C3b to form an inactive C3b and release a part of the C3b, which is C3F. Now, this C3b inactive form can be further degraded by factor I with the help of a cofactor to form C3C, C3DG, etc. And this can never bind to the factor B. So you cannot have, once you have the C3Bi formed, this cannot go back into the C3b feedback loop at all. So therefore, what you observe is that the C3b is getting degraded by the C3b breakdown loop, and here what you see is more and more of C3b getting formed. So this is an important regulatory mechanism that is observed, and you can see over here how factor H and factor I along with different cofactors are responsible for ensuring that the levels of C3b are maintained. So factor H is the soluble fluid phase initiator. That means it is present in circulation and it can inhibit the alternate C3 convertase. Factor I is again a serine protease that cleaves down C3b into C3bi. This is inactive and it cannot bind to factor B, so you cannot see further amplification of C3b. Factor I is proteolytic. Now, factor I is not really, in an inactive form, that it is not a zymogen. It is already a catalytic form itself, but then it is in an inhibited form. So when you have cofactors coming and binding to the factor I, then it becomes active. So these cofactors are membrane cofactor protein, decay acceleration factor, complement receptor 1, and these are also present in the circulation. Now let us come to the fact that alternate pathway is not going to initiate lysis of the host cell. So let us try to understand how the alternate pathway is regulated in order to ensure that the host cell is not lysed. So say for example, you have the resting or the activated host cell. When we say activated host cell, it means the C3bb has come and bound, and then you have factor I coming and binding to convert the C3b into an inactive form. And this inactive form is further converted to C3C and C3D, which can never go and bind to factor B at all. So you can see how factor I is ensuring with the cofactors, of course, factor H and the cofactors is ensuring that even if C3B is bound to the surface, it is not getting activated. It is only going to get further inactivated and broken down into different components. That is one. What can also happen is that the cofactors, no, like for example, the CR1, the DAF, and the MCP, along with the factor H, can come and bind to the C3BB and stop its activation. How? So here is a case in point. So the factor H can actually bind to the C3B in such a way that it does not allow the C3B to get active. Now, this binding of factor H to the C3B is because the factor H has several domains that can bind to glycosaminoglycans that are present on the surface of the host cell. Now, say for example, many of the host cells have sialic acid. Microbes do not have sialic acid specifically. So, factor H doesn't really go and bind to the microbes. So there, if the C3B is bound to the microbes, you don't have the factor H and the factor I actually going to a pathogen that is bound to C3B. It will only come to the host cell. Why? Because the factor H can recognize specific glycosaminoglycans that are present in the factor in the host cell, and because the factor H can come and bind to the C3B and the host cell per se, it is going to ensure that the factor B does not even come and bind to the C3BB. So the question of the C3B getting active doesn't arise. So this is something that is important, and this is something that kind of happens. So we basically are therefore understanding that the C3B on binding to pathogens may not be inhibited by factor H and factor I, but the C3B that is bound to the host cell can be inactivated by factor H and factor I along with the cofactors MRP, DAF, and the CR1. Now, what happens after the C3BB is formed? Okay, so the C3BB, once it is formed, we already know that it is acting as C3 convertase. So it is going to form C3B, more of C3B. Now, C3B on being formed again can bind to C3BBB to form a complex which is called as C3BBB-C3B. Now, this complex is acting as a C5 convertase, and this will now convert C5 into C5A and C5B. So, you're basically looking at the fact that the C3BB-C3B complex, which is the C5 convertase, can bind to C5 and convert C5 or cleave C5 such that you get one component which is C5B and the other component which is the C5A. C5A is an anaphylatoxin, and therefore it will move away and act as a mediator of inflammation, while C5B becomes a molecule that can bind to the membrane of the pathogen, to which then you can have binding of the C6, C7, C8, and C9 to form finally the membrane attack complex. And we know that with the formation of the membrane attack complex, the osmolarity of the cell is affected. There is a flushing in and out of components happening, and therefore the cell loses its integrity and ultimately it lyses and dies. So therefore, elimination of the pathogen through alternate pathways also by ultimately forming the membrane attack complex. But the point to be noted is that the starting point or the starting molecule for alternate pathway is C3. Okay, and C3 will bind to C3BB and C3B again to form a C5 convertase, and thereon, once you have the C5B formed, we know that a membrane attack complex can be formed. So let us make the conclusions. The serum C3 can spontaneously form C3H2O, which binds with factor B. This complex is then acted on by factor D to form C3BBB, which is a serine protease. The C3BB is stabilized by properdin and thus can leave more C3, cleave more C3 to form C3A and C3B. So effectively, there is an amplification of C3B that is going to happen. This C3b can further bind to factor B to form more of C3b, and that is what is creating an amplification loop for formation of more C3b. The C3b can bind to pathogens, but they can also bind to host cells and to apoptotic cells. So under physiological conditions, its ability to bind to apoptotic cells ensures that damaged tissues or damaged cells are eliminated because the C3b binds to it, and it calls in different processes to ensure that the apoptotic cells are silently removed. But when it binds to the pathogens, the C3b is able to initiate the alternate pathway to ensure that a membrane attack complex is formed on the pathogen and the pathogen is killed or lies. But that it is able to bind to host cells, that it is able to bind to apoptotic cells, that it is able to bind to pathogens, there has to be a regulatory mechanism to ensure that it does justice to whatever it binds. So factor H is a soluble inhibitor of the alternate pathway C3 convertase. What is this? It is this, okay, and can degrade C3b. So factor H is responsible for degrading C3b. But it does not directly degrade C3b. It degrades C3b with the help of factor I. And factor I with its cofactors can degrade C3b, especially on the host cells. So alternate complement pathway has its role in physiological conditions as well as pathological conditions, prompting its regulation, and and it has been observed that an inappropriate immune responsible is possible if there is no regulation. Thank you.